Hi there. This is a short video to deal with the AS topic, exchange and transport, which is a module 2 topic. If you're watching this video, you probably already have a set of my notes and a set of long questions and a set of short questions and the answers to all those questions. So I haven't planned this too carefully, but essentially what I want to do is go through this topic fairly quickly and deal with the main parts of it and try to go through the answers to some of the main questions. So as I say, to get the most benefit from this video, you really need to have all these resources in front of you. If you don't have these resources in front of you, you can get in touch with me there by mobile, and I'll get some resources to you, and then you can benefit more from the video. Okay, so there's quite a difficult AS topic, quite a lot of different elements to it. So I'm going to try and go through the major elements, and I'm not going to go through all the questions in the question booklet. I'm just going to select particular questions which are maybe more recent questions and questions that are dealing with the main parts of the topic. So where are we going to start here? Well, I'm just leaping into my note booklet here and just essentially following the order of the topics as they're found in the note booklet here. Essentially, the first thing that comes to my notice there is the topic of surface area to volume ratio. What is this topic essentially about? This topic is essentially about ways in which molecules and substances are transferred into and out of organisms across surfaces, across membranes, across cell membranes. So we're essentially, when we think of exchange, we're thinking about gases like oxygen and carbon dioxide, but we have to consider also other things like exchange of water across surfaces and ions, mineral ions, for example, across the surface of the cells and the root of a plant. That's exchange as well, and so on. So we're dealing with and also, for example, the movement of the the movement of food molecules, digested food molecules, across the wall of the ileum. We don't have to look at any of those things in great detail, but we have to be aware that we're dealing with the movement of substances across surfaces in and out of organisms. So essentially, the main aspect of this topic is the movement of the gases oxygen and carbon dioxide across the alveolar surface in the lungs of mammals, and a little bit then on the movement of gases in and out of plants across the spongy mesophyll surfaces on the inside of leaves. So those are the main areas. So essentially, I'm going to work my way through the notes here as quickly as possible, and as I come to each topic, we'll have just a wee bit on each topic, and then I'll maybe look at appropriate questions. So first of all, the idea of surface area to volume ratio. Well, this generates questions quite frequently. The idea of surface area to volume ratio. Now I can draw diagrams on the screen here, but if I start to do that, it'll simply take me too long to get through this topic. And I probably intend to spend less than an hour. So what you need to do is you need to have my notes in front of you, because the diagrams are all done really well in the notes. So I can refer you to the diagrams. So I'm looking here at page 3. I'm looking at this idea of surface area to volume ratio. And near the bottom of the page, I discuss the idea of surface area. The surface area of an organism is a measure of how many cells of the organism are in contact with its surroundings. The more surface area that an organism has in contact with its surroundings, the more surface there is for the uptake, for example, of the gas oxygen into the organism and the release of the gases gas carbon dioxide from the organism. So the surface of an organism is important then in terms of, because the surface is in contact with the environment, contact with the air, and you want the maximum surface possible to take in oxygen. So if you think about this, for example, a very small organism that lives in water, then it might be taking in oxygen from the water around it. If it's a very small organism, then its surface, the surface of the organism is all it has to depend on to get oxygen into its body. So its surface area is pretty important. What's the significance of the volume of the organism? Well, the volume of the organism represents the volume of cells that are present in the organism, the volume of cells which require the oxygen that's been taken in by the organism. So if you have an organism, then its surface area gives a measure of the surface available to take in oxygen, for example. And the volume is a measure of the number of cells in the organism that need oxygen. So what do you want to maximize? You want to have the maximum surface area of the organism so that it can get sufficient oxygen to all of its cells. But we want to now look then at how the surface area and the volume of an organism changes as the organism gets bigger. And this is usually done with cubes, and I'm looking here at page 4. This is usually done with an organism that's shaped like a little cube, because it's easy to work out the surface area and easy to work out its volume. On the bottom of page 4 then, I'm looking at an organism, let's say it's living in water, and it's got a side of 2 by 2 by 2, 2 centimeters by 2 centimeters by 2 centimeters. You can see its surface area there is 24 square centimeters. If you work out its volume, so its surface area is the surface across which it might take in oxygen. If you work out its volume is 2 by 2 by 2, which is 8 cubic centimeters. So it is 8 cubic centimeters of cells that require oxygen and 24 square centimeters of surface to supply oxygen. We can calculate something called the surface area to volume ratio. So that's simply the surface area up the top divided by the volume. So the surface area would be 24 divided by the volume, which is 8. So the surface area to volume ratio is 3. In other words, for each cubic centimeter of cells, there are 3 square centimeters of surface to get oxygen to those cells. Now, if we compare that with a larger organism then, and I've just made an organism there on top page 5, it's twice as big, and it's got a side of 4. You work out its surface area and volume ratio, surface area and volume there, you'll see that its surface area is 96, which is, by the way, sorry, I need to say that there's 6 sides to any cubic organism. So therefore you just work out its surface area of one side, which is 16. 4 before 16, 6 16 is 96 square centimeters. So its surface area is 96 square centimeters. So if you compare it with the smaller organism, you can see that its surface area has gone up by 4 times. So it's got 4 times more surface than the smaller organism with a side of 2. 4 times more surface. So it's got more surface to get in more oxygen. But if we measure its volume, work out its volume is 4 before, which is 64. So if you compare to the smaller organism, its volume has gone up by 8. So note there that its volume has increased by 8, but its surface area has only increased by 4. So its volume's getting bigger, surface area is getting bigger, but the volume is increasing much more quickly than the surface. In other words, the surface area isn't keeping up with the volume. So as organisms get larger, or if you have larger examples of the same organism, have a lower surface area to volume ratio. The bigger organism there, you can see if you calculate the surface area to volume ratio, it's 96 over 64, which is 1.5. In other words, the bigger organism only has 1.5 square centimeters of surface for every 1 cubic centimeter of volume of cells. So it's better to be a smaller organism, because a smaller organism has got more surface relative to its volume. And once you get bigger, then your surface may not be able to keep up with your volume, and there may come a point where your surface is not sufficient to supply sufficient oxygen to all the cells. Just out of interest, there's another aspect there. If we consider that the oxygen is diffusing through the surface of the organism, it's diffusion we're talking about here, then for the bigger organism, the cells in the very center are further away from the surface. So it may be more difficult for oxygen to diffuse all the way to the center of the organism. Whereas the smaller organism, all the cells in the smaller organism are close enough to the surface to get oxygen. So in general, as organisms get bigger, their surface area to volume ratio decreases. And you often get this represented in a graph, or maybe even in figures, as on page 6 there. You can see that, there's an inverse relationship. So conclusion from all of that is that if you're a very small organism, and for example, living in water is a good example, very small organism living in water, then you may have sufficient surface to get you sufficient oxygen to supply all your cells. Whereas as you deal with bigger versions of that organism, may not have enough surface to supply oxygen to all their cells. And there may come a point with the size of the organism where the surface is not sufficient. The surface is not sufficient at all. It won't supply sufficient oxygen, in which case you have to, the organism has to develop or evolve a specialized gas exchange surface. So take us, for example, our surface area, which is our skin, our surface area to volume ratio will be very low. We simply don't have enough skin on our surface to breathe through, even if we could breathe through our skin. So our surface is not big enough. So we've had to revert to developing a specialized gas exchange surfaces inside our lungs. And essentially that is essentially our lungs, and the gas exchange surfaces there are the walls of the alveoli. And there's such a large number of these alveoli packed inside each lungs that there's something in the region of 70 square meters of surface. It's a bit like having 70 square meters extra surface packed away inside you, through which you can get oxygen. And 70 square meters, that's quite a lot. 1 square meter. At school we were told that 70 square meters has something to do with the possibly the size of a tennis court. I don't know whether that's actually the case, but I always remember that from school. We've got the surface area of a tennis court inside our lungs, inside our chest cavity, through which we can take in oxygen. So there's another aspect to this as well, that you take in oxygen obviously for respiration inside your cells. If you're an organism that's very active, then you need more oxygen to supply the extra energy needed for respiration. So for example, a small organism that's got enough surface to supply oxygen to its cells whenever the organism is at rest, that organism may not be able to be active, because to be active it needs more oxygen for more respiration, and to be active there it just simply doesn't have enough surface to get sufficient oxygen in there. So I think that's what generated that question there on page 7 in the notes, at the top of page 7. It's a suggest question. Suggest why a mouse might need a bigger gas exchange surface than a frog. By the way, a mouse has a set of lungs. A frog also has a set of lungs, but a frog also breathes, it does some gas exchange across its skin. So good question, how does a frog breathe? It's got two. It's got lungs, but it's also got the surface of its skin. And an interesting point about that is the surface of the frog's skin must always stay wet if it's to get oxygen across its skin. And this is a general idea that you need to know, that for any gas exchange surface, the gas exchange surface must be wet. And the reason is that for oxygen, for example, to cross a surface like the skin of the frog, the oxygen has to dissolve in water first of all. So that's a very important point. Oxygen must dissolve in water before it can diffuse across, it can move across a gas exchange surface. And that applies also to the alveoli. Oxygen inside the alveoli has to cross across the very thin wall of the alveoli, but it must dissolve first of all in a layer of moisture which is on the inner surface of the alveoli. Another example would be an earthworm. An earthworm is small enough that it doesn't need any lungs or gills or any kind of gas exchange surface. So it's a good question. How does an earthworm breathe? Actually, it breathes through its skin only. It relies completely on its skin. In other words, an earthworm, because it's long and slender, it's got a very large surface area, relatively large surface area compared to its volume. Enough surface area that it can get sufficient oxygen in through its skin. But being as its skin is its gas exchange surface, the skin of an earthworm must always stay wet. It must stay wet. If it doesn't stay wet, the earthworm can't breathe through its skin, and the earthworm then dies. Okay. So just to come back to the question about the mouse and the frog, I suggested here that the mouse maybe, well, two things about the mouse. The mouse is what we call warm-blooded. In other words, the mouse maintains a high body temperature, and to maintain a high body temperature, it requires to do a lot of respiration in its cells. So therefore, it needs more surface area to bring in sufficient oxygen to supply for the extra respiration to take place in the cells. The mouse may also be more active. Maybe if an organism is more active, moving around more, then it needs more oxygen for more respiration. Now, in relation to surface area to volume ratio, there isn't usually a calculation question. You don't usually get calculation questions, but question 3 there in the booklet is a good example of a fairly recent calculation question. I think it's been 3 or 4 year old, and it's quite difficult, but it's not. It's quite difficult to some extent because it involves a bit of maths, but it's not too bad. I'm not going to go through the whole question because you have the answers. If you have my booklet here, you also have the answers to the questions. And if I go through all the questions, I'm not going to get very far in this topic. So calculating the surface area of this small organism here, it's got a side of 12 micrometers. So you work out its surface area. So the surface area is 12 by 12, which is 144 by 6. Gives 864 micrometers squared. That's its surface. And then its volume is 12 by 12 by 12, which comes out at 1728 micrometers cubed. So you've got its surface, a surface area, and it's got its volume. And they could have asked you then for its surface area to volume ratio, in which case you just put 864 over 1728, and that'll be its surface area to volume ratio. They didn't ask that. Then they told you then, when this organism is at rest, that's an important thing. You want to underline that. When this organism is stationary and not moving around, it absorbs oxygen at the rate of 0.02 micrometers cubed for every micrometer squared of its surface. You understand that then? So micrometers to the -2 means per square micrometer. So it absorbs oxygen at that rate, 0.02 per square micrometer. And then down below it asks you how much oxygen does it absorb. So you have to multiply the 0.02 by the total surface area. So it's 0.02 times 864, and that comes out at 17.28. So that's how much oxygen it's absorbing in micrometers cubed, the volume of oxygen per minute. Oxygen consumed, well, it tells you that it consumes 0.01 micrometers cubed of oxygen per micrometer cubed of its volume. So its volume was 1728, So you multiply 0.01 by 1728, and you get your answer. Oxygen consumed at rest comes out at 17.28 micrometers cubed again. And that's a bit of a surprise because doing our calculation, we've worked out that the amount of oxygen absorbed is exactly equal to the amount of oxygen consumed or being used up with this organism. Whilst it's at rest, the amount of oxygen it's absorbing is exactly just the amount of oxygen that it needs. And then it says, why must this organism change its shape if it's going to become active? Well, the way it is at the minute, it doesn't have any extra. To become active, it needs to do more respiration. To do more respiration, it needs more oxygen. At the moment, it can't get any more oxygen. The amount of oxygen it's taking in just meets its needs at rest. If it's to move, then it's going to need extra oxygen. So it must change its shape if it's going to become active. And you would say something like, to become active, the organism needs to do a greater amount of respiration, needs to do more respiration, so that'll require more oxygen. Where's it going to get more oxygen out of? Or if it could change its shape, so if instead of being a cube shaped, let's say the organism, you were to squash the organism so that it became flat like a pancake. Let's imagine it could change its shape so it was flat. Then it would increase its surface area. And by increasing its surface area, get the extra oxygen it needs for the extra respiration, for the extra energy. So that's the 2 marks for that. You can check out the precise marks, the way it's written in the mark scheme there. Why smaller specimens of the species are more active? Well, smaller specimens have a larger surface area to volume ratio. That's their first mark. And you see, because they have a larger surface area to volume ratio, they can get the extra oxygen required over and above that needed just for their normal respiration, the extra oxygen required to allow them to move around. to move around. So that's a surface area to volume ratio question. Quite difficult. The part one there, A part two was very difficult for biology students, especially because they're not so used with micrometers and those sorts of values. Question 3 is also a surface area to volume ratio type question 2. Sorry, question 4. My mistake. I'm not going to go through it. It's quite difficult. But you've got the answers there, so I think that's maybe enough surface area to volume ratio questions, maybe at the moment. So question 4. By the way, any of these questions that you still don't get to the bottom of, we can maybe Skype on them at some stage, and we can get to the bottom of the more difficult ones. Question 4, by the way, was about 6 or 7 years old, that question. So surface area to volume ratio, hopefully you've got some idea of that then. Okay, just running through the notes then, you'll see that the next pages 8 and 9, pages 8 and 9 are just general examples of situations where you have large surface areas for exchange. In the villi, you have a large surface area for, in the villi, in the ileum, you have a large surface area for the exchange of digested food between the lumen of the ileum and the capillaries in the villi. So large surface area created by villi. Root hair cells from GCSE have an extension which gives them extra surface area for the uptake of water and ions such as nitrates by active transport and so on. There's just an example of a small organism it's a flatworm, with its adaptations for more rapid uptake of nutrients or water from its surroundings. The flatworm lives in water, and you can see it's got a large surface area because it's a very flat body. It's leaf-like in shape. So most of that is self-explanatory. On to page 9, alveoli. Alveoli then are gas exchange surfaces inside our lungs, and frequently they'll ask you about alveoli. The large surface area, well, first of all, they have a very large surface area, and sometimes they'll ask you, how do you get this large surface area? And how do you get a large surface area created there? And you should say something, there's millions of alveoli. I think I've written there large numbers of alveoli. If you want to write above that millions, the examiners, if you ever get the question, that's the examiners would prefer you to write millions rather than just large numbers of alveoli. And red blood cells then also have large surface then to get oxygen into the hemoglobin. So these are minor aspects here just for the moment or two. Turn the page then, thin surfaces, any surface across which materials are moving need to be as thin as possible. So you have alveoli are 1 cell thick, have walls of 1 cell thick. The walls of the villi are 1 cell thick as well, and walls of capillaries are just 1 cell thick because capillaries allow exchange of oxygen from the blood to the cells through which the capillaries are flowing, exchange of oxygen and other things. Concentration gradients. Well, concentration gradients, you'll come across even in GCSE, this idea that the rate at which a substance moves across a surface will depend on the concentration of the molecule or the substance either side of the surface. And what you want is the biggest difference in concentration. So if you have a very big difference in concentration of oxygen between the inside of the alveoli and the capillary, which is wrapped around the alveoli, the blood, then you get the maximum rate of diffusion across that surface. So a big diffusion or concentration gradient is important. Now, there's some aspects here that will be dealt with later when we come to look at the lungs. The next thing I'm going to tell you about there is this idea of mass flow. Mass flow, transport of substances inside organisms, and you come across this idea of mass flow. So once the substance, for example, is inside the organism, then how might it move around? So mass flow is just a description of several mechanisms within organisms where a substance, for example, air with oxygen in it, or water or blood, is moved around inside the organism. and that movement is brought about by pressure changes. So a definition of mass flow, it has never been asked, but a definition of mass flow is the movement of the transport of a substance, bulk transport of a substance, for example, blood, around an organism, usually inside pipes or tubes. In the case of blood, it's arteries and veins, brought about by a pressure difference or pressure gradient. In the case of blood, it's the high pressure that's exerted by the contraction of the ventricles, which creates a very high pressure and pushes blood around the body. So there are 4 examples there you'll see on page 12. 2 examples which come from mammals, from humans, for example, and 2 examples that come from plants. And in each case, note the key thing that I've underlined there, the idea that there's a high pressure, created. You get a pressure difference or pressure created. And, and also that the bulk flow of material through tubes or pipes. Okay, we'll talk about those a wee bit. We'll talk about blood, and we'll talk about that in that topic, blood, heart and circulation, which is at AS2. And we'll talk about the mass flow of air through around the lungs. We'll talk about that later on. So now over the page, we're going to talk then aside about gas exchange. So we're going to concentrate purely on the idea of gases. And the movement of gases across surfaces is described by something called Fick's law, F-I-C-K-S, Fick's law. Not really a law, it's really just a set of description of how the rate of a gas, for example oxygen, would move across a surface. How the rate of a gas moves across, what factors affect it? What is it that speeds it up or slows it down, the diffusion of, for example, oxygen or carbon dioxide across the walls of the alveoli, across the surface. Fick's law basically says that the rate of diffusion of the gas is proportional to the surface area. First of all, in other words, the bigger the surface area, the faster the rate of diffusion. So the more surface there is, the more oxygen can diffuse. The concentration difference or the concentration gradient. So it's proportional to the surface area. It's also proportional to the difference in concentration. So the bigger the difference in concentration of the gas either side of the surface, and it doesn't have to be a gas, it can be any other substance across the surface, the bigger the difference in concentration and the faster the movement of the substance. And that sort of throws up the idea that if the concentration of, say, oxygen was the same on either side of a surface, you get no diffusion at all. You need a difference. The bigger the difference, the better. And the third thing is the thickness of the gas exchange surface. The thickness, so the rate of diffusion goes up, increases as the thickness goes down. In other words, thin gas exchange surfaces are better for a fast rate of diffusion. So it's a simple equation. Simple, it's not really an equation. I would just write it like rate of diffusion is proportional to the surface area up the top, concentration gradient up the top, and down the bottom of the equation put the thickness of the diffusion surface, or the length. Sometimes it's called the length of the diffusion path. In other words, how far does the substance have to diffuse. Now, questions have been asked about that then. Let's see, we look at the question again, just see if there's a, you sometimes just get asked to write down Fick's law. Other times you get asked to apply it. Now, so we're going to just quickly look at question 6, which is, I think this is possibly, you can write at the top of question 6, 2019. I think this is the most recent question. The most recent question on the gas exchange in this book or two. Sorry, most recent question in from CCEA, module 2. Question 6 then. So it starts off by asking you to say what Fick's law is. So you have to write down your Fick's law at the start, and you get a mark for that. And then it said, apply Fick's law to state Fick's law and describe how gas exchange structures of plants and mammals are adapted to maximize diffusion of gases. Your answer should also be, so then you have to talk about the effect of smoking and damaging your lungs, and how that slows down the rate of diffusion. Now, to to answer this question, I think, without spending too much time on this essay, because essentially the answer to this essay is in my notes. If we go to page 8 in the question booklet, page 8 in the question, sorry, 8 in the note booklet, my mistake. Page 8 in the note booklet, and this is page 8 in the second half of the booklet. By the way, this booklet split into two halves. The first 12 pages, and then we start again on page 1, gas exchange. So it's quite near the end of the booklet's question. It's page 8 and 9. Gas exchange in mammals, part C. It's basically looking at the, first of all, we're saying the gas exchange surface in mammals is the alveoli. Alveolar walls, the walls of the alveoli. And this essay then was asking you to discuss, describe gas exchange structures in mammals and how they're adapted to maximize the diffusion of gases. And you can see there's 15 marks there. So just splitting that down, essentially we're talking about 5 marks for alveoli and lungs, 5 marks then for the gas exchange structures of plants. Okay, we'll come to that in a minute or two. And approximately 5 marks then for how smoking damages the gas exchange surface, damages the rate of diffusion, basically damages your ability to diffuse oxygen and carbon dioxide in and out of your alveoli. Now, to answer the bit about the alveoli, first 5 marks, I'm just looking there on page 9 in my notes, and essentially I'm pointing you to this because I don't want to go through all this in great detail, otherwise we'll not get this topic done. But essentially there on page 9 you'll see a picture of the alveoli there at the top, and then underneath underneath the second half of the page 9, it says, what features of alveoli maximize diffusion rate? What features of alveoli maximize diffusion rate? And essentially, those are essentially what you need for your description, your explanation in that essay. So you've got 1, 2, 3, you've got 8 points there, and if you look at the mark scheme for that, don't immediately have the mark scheme at hand, and you just look here and see the total number of marks. So see in the mark scheme there that I supplied, you'll see that it's question 6. You'll see that I've numbered the marks off. First of all, number 1, that was the fixed law answer. And then 2 to 7. 2 to 7, that gives you marks to do with leaves. We'll leave that first of all. And then the second 8 marks, 1 to 8, and the second third of the answer scheme there. Those are the marks that they allowed. You can see they were quite generous there. This whole essay needed 15 marks, and in total they gave 7, and it's 15, 10. They have 24 points, which is very generous. And basically they allowed you, they didn't specify where you had to get your points from. They basically said any 15 points out of 24, that's considerably generous in terms of our mark scheme. So you'll see that there's in the middle of the answer scheme, then 1 to 8, essentially that's essentially those points I have there on page 9. So that describes, you'll see there millions of alveoli, large surface area, spherical shape of the alveoli. Moisture on the surface of the alveoli allows oxygen to dissolve, so very said about that. Dense capillary network wrapped around the alveoli. There's no point in having alveoli with large surface area if you don't have a lot of capillaries in contact with those alveoli. So traditionally you would just show an alveoli looking like this, and then you would suggest, the teacher would suggest that there was a capillary lying close to the alveoli, and in here you'd red blood cells, and they would suggest there was just one alveoli and one capillary. That's not the case. In fact, you'll see if you're looking there on page 8 in my notes, you'll see there that the alveoli are crisscrossed with hundreds of capillaries all going across the alveoli. So there's loads and loads of capillaries all stretched, all wrapped around the alveoli. And the idea is that you want the maximum surface area of capillaries in contact with the walls of the alveoli, because then you have maximum surface area across which you can get oxygen diffusing. And dense, so a mark for a dense capillary network around the alveoli increased surface area. Squamous endothelial cells of the alveoli. And so you get a mark there for saying that the cells that line the alveoli is just one cell thick. The surface of the alveoli is just one. I didn't do that very well there. And so the surface of the alveoli is just made of a single, I'm just putting some nucleus there, a single layer of squamous endothelial cells. And they needed the squamous endothelial cells. They wanted to know that word, squamous endothelial cells, one cell thick. Or you could also have said that the walls of the capillaries are just one cell thick, one layer of squamous endothelial cells. Red blood cells can squeeze through. The capillaries are so narrow that the red blood cells have to distort and shape and become long and skinny to slide through. And as they slide through, they're pushed tight up against, the red blood cells are pushed tight up against the walls of the capillaries, so that you're minimizing the distance from the inside of the alveolus into the blood, into the red blood cell. They move slowly. The red blood cells move slowly. They move relatively fast, but slow enough so it gives enough time for diffusion. And ventilation. Ventilation is just a fancy word for breathing in and breathing out. So ventilation is made up of inhalation, exhalation. So too many words for breathing in and breathing out. Inhalation is also called inspiration, and breathing out or exhalation is also called expiration. Anyway, ventilation is breathing in and breathing out. It says there ventilation maintains a high concentration gradient. Well, ventilation ensures that there's always plenty of oxygen present inside the alveoli. So that the oxygen gets used up inside the alveoli as it moves across into the capillary. But ventilation, breathing in and breathing out, ensures that there's always plenty of oxygen there. So that maximizes the diffusion gradient. Okay, so that's the mammalian aspect of it, our lungs. The other aspect they wanted to know about, well, they want to know about smoking. So let's just briefly look at smoking. You look on page 11 there. That summarizes quite well the effect of cigarette smoke on smoking. On breathing and on gas exchange in the lungs. So there's quite a lot of stuff there. If you look through the mark scheme in the essay, I think they give 9 points there, and you'll find that they're all contained on that table on page 11. And then they wanted also, so this is quite a comprehensive essay. You wanted you also to talk about gas exchange in plants. So if you turn back to page 5 there, and there's a picture of a leaf at the top of the page. And it's asking then, it wants to know, by the way, the essay is asking you at all times to relate Fick's law to the gas exchange surface. So it's wanting you to talk about how you get large surface area, how you get a good concentration gradient, and how you minimize diffusion distances. So you have to relate those to the lung, to the alveoli. And now you also have to relate those to plants. Plants are considerably more difficult. The gas exchange surface of a plant, at GCSE, if somebody had asked you what's the gas exchange surface of a plant, you'd have said the leaves, possibly. You've been quite good, that'd been quite good. But it's not the leaves, not the surface of the leaves. Upper surface of the leaf got a waxy cuticle. Gases can't get through that. The lower surface, however, has stomata. But that's not the lower surface is not the gas exchange surface. It's actually inside. Once you go in through the stomata, you go inside the leaf, you go into a layer called the spongy mesophyll layer. And then above that, you've got the palisade mesophyll layer. Essentially, it's the surfaces of, once you go inside the stomata, it's the surfaces of the inside of the leaf of the spongy mesophyll layer. By and large, that, and the surfaces of the spongy mesophyll cells is the gas exchange surface. So if you had oxygen trying to make its way into the cells of the leaf, the oxygen would diffuse in through the stomata into the air spaces, and then it would dissolve on the liquid, the water on the surfaces of those spongy mesophyll cells. So note once again that the surface across which diffusion takes place must be wet. So the surfaces of those spongy mesophyll cells will be wet. And you'll see there I've given about 6 marks there, or 6 points in my notes on page 5. So spongy mucosal cells have a large surface area. Stomata open to allow gases in and out. So you talk about how gases get in and out there. Leaves are thin, giving a short diffusion distance. So once the gas diffuses in through the stomata, once oxygen or carbon dioxide, it doesn't really matter, either one of them you can refer to it, once they diffuse in through the stomata, then because the leaf is thin, it means the distance from the bottom of the leaf where the oxygen and carbon dioxide go in to the top of the leaf is very small. That means oxygen doesn't have, it only has a very short distance to travel to get to all the cells in the leaf, because all the cells in the leaf are near the bottom. That makes sense, very thin leaf. So, but you have to specify that in your answer. You can't just say the leaves are thin. The full answer would be to say leaves are thin, therefore there's a short distance from the stomata to all the cells. That means a short diffusion distance, therefore that maximizes the rate of diffusion. Fick's law, referred Fick's law here again. Stomata open during the day to allow maximum amounts of gases inside the leaf. That maintains a high concentration gradient. The last point there, photosynthesis uses up carbon dioxide and respiration uses up oxygen. If you get inside a plant, inside a spongy mucosal cell, if you get all the carbon dioxide being used up for photosynthesis, then there's a very low concentration of carbon dioxide inside the cell. If there's low concentration inside the cell and there's a high concentration outside the cell because it is diffused in through the stomata, then you've got a concentration difference, a concentration gradient. So you get a mark for saying that, maintaining concentration gradient. Okay, as I said, all the stuff about the smoking, essentially the marks of the smoking, page 11 there. Now, what else? That's a pretty important question there, very recent question. I'll maybe come back and look at question 7 in a moment or two. It's quite difficult about surface area to volume ratio. Where do we want to go now? Yeah, just how we looked at the ideas of inhalation and exhalation and mass flow. So you'll see in the second half of the question that question 1 is asking you mass flow is a term given to transport mechanisms which involve the use of a force. I would have said the use of pressure differences or pressure to move large amounts of substances. Ventilation in mammals is an example of transport involving mass flow. And you have to describe, well, without, they're going to ask you how does mass flow of air out of the lungs is achieved during expiration. Let's just first of all say that the movement of air in and out of the body, breathing in and breathing out, is an example of mass flow. Why? Because you create pressure differences due to your lungs, due to your intercostal muscles and your diaphragm, creating high and low pressures that move the air in and out through your pipes, through your trachea and your bronchi and out of your mouth and so on. So it is an example of mass flow. But essentially this question is just asking you to describe expiration. So you look at the notes there. I don't want to spend too much time on this. Page 10 in the notes. Ventilation of the lungs. Ventilation is the mass flow of air in and out of the lungs brought about by pressure changes. Ventilation ensures continuous high levels of oxygen inside the alveoli, so that a high concentration gradient exists for oxygen between the alveolar space and the blood in the capillary. And there you have descriptions of both breathing in and breathing out. Breathing in, inspiration, breathing out, expiration. And that's essentially what your 4 marks are for there for expiration. So essentially you could write down the 4 marks as they're written there. You could say for expiration, what happens during expiration? You'd say expiration is breathing out. You'd say diaphragm and intercostal muscles relax. Diaphragm and intercostal muscles relax. And once they relax, then the volume of the air inside the lungs decreases as the diaphragm raises up and the ribcage falls. So the volume decreases. That causes the pressure in the thorax to rise, and that pushes air out of the lungs. So those are the 4 marks there. Get those 4 points. For breathing out, and inspiration is the opposite of that. Question 2 there, just a question about the lungs. Question 3, Fick's law, quite an old question. How are the above factors achieved by increasing the surface area of the membrane and increasing the concentration gradient across the membrane? How are the above factors achieved by mammalian gas exchange surface? So the mammalian gas exchange surface is the alveolar walls. Surface area, how does it increase? You have to say there's millions of alveoli, and you would say also that they have a rounded or globular shape. I'm trying to think of what other word, spherical shape. You get a mark for that as well. You'd say spherical shape of the alveoli increases the surface area. You'd also say there's also a large surface area of capillaries in contact with the alveoli, so you get a large surface area. Concentration gradient increase. You could say ventilation. Ventilation ensures there's a very high concentration of oxygen in the alveoli all the time, because you're constantly replacing any oxygen that moves out of the alveoli. And the other thing is you could say that the transport of the oxygen away quickly here ensures that the concentration of oxygen in this region in the blood is always kept low. So you're keeping a very high concentration in here due to ventilation. So ventilation is constantly bringing lots of oxygen in there. So you get lots of oxygen there, and the blood's quickly moving the oxygen away. So there's always a big difference in oxygen concentration between inside the alveoli and in the blood. There's always fresh blood arriving with no oxygen. That maintains high concentration. Name one other factor increase the rate of diffusion across a membrane. A thin membrane. So in this case you could say the alveolar walls are just one cell thick. The walls of the capillary are just one cell thick. How is this factor achieved in mammalian gas exchange? You could say those two. Emphysema and bronchitis. Well, emphysema and bronchitis. Sorry, I'm rushing here, but I'm just trying to get as much covered as possible. Emphysema is a disease brought on usually by smoking, and it's a disease where your alveoli are damaged by smoking. Typically, alveoli may be drawn like this, like a series of like bubbles, like a bunch of grapes sometimes, like that. So that's the normal alveoli. Now, and so you've got quite a lot of surface area. So actually alveoli sometimes are just drawn at GCSE like that. They're not really like that. They're more like that. More like a bunch. Bunch, they got a bunch of grapes. Now, and you've got quite a lot of surface area there. All that surface. So now, what an emphysema, tar, which is an aspect of cigarette smoke, destroys the walls of the alveoli. So it kind of removes a lot of the walls of the alveoli. It makes the alveoli sort of almost coalesce into one alveoli, one big alveoli. So you can see how they've reduced the surface area of the alveoli. So by reducing the surface area, I'm just reading from page 11, the very bottom. Tar causes the breakdown of the alveolar walls, reducing the surface area for gas exchange. So that will answer that question. And bronchitis then. Bronchitis is inflammation and infection in your bronchi, and brought about by the presence by cigarette smoke as well, because the tar damages and causes it inflames or damages, irritates the walls of the bronchi, and that causes a process called inflammation, where the walls of the bronchi swell, so that the bronchi pipes become narrower. And if you get a narrower pipe, then that slows down the ventilation rate. It means less air can get into your lungs. So the bronchi narrow due to and the buildup of mucus also, which is normally there and is normally useful because it traps bacteria and makes the walls of the alveoli moist. But if you get inflammation due to damage, then mucus builds up, so that the walls of the bronchi pipes become even covered in all this thick mucus, and so the lumen becomes smaller and smaller. So there's less area less lumen, less opening for gases to move in and out. So that reduces the ventilation bit. Now, so I'm really just tidying up one or two more questions here. You have, as I say, you have all the answers there. Question 4, Fick's law, essentially the same. Fick's law states the rate of diffusion increases the concentration gradient is high, under the diffusion gradient distance is short. So once again, you have to see how you get a high concentration gradient and how you maintain a short diffusion distance. All that's pretty straightforward. Part 3, there's a third factor, which would be so surface area, concentration gradient, and the thickness, the short diffusion distance. Part B, that surfactant, special cells around the alveoli secrete a substance called surfactant. So I want you to just have a look then at this surfactant thing. Sometimes it gets asked about. So if you go to question 9, sorry, go to page 9 in the notes again, at the top there, it says a wee bit about, it shows you a picture of an alveoli. And I just highlighted 3 types of cells that are present in the walls of the alveoli. And one of them, the first one there I've said surfactant secreting cells. So that's what this question's about. And if you just read the question, it says some babies are born without enough of this surfactant. So this is a condition of premature babies. Premature babies often need assistance to breathe, and it can be due to the fact that their lungs haven't grown sufficient, they haven't developed enough, and they don't make enough of this surfactant stuff. And you'll see what the surfactant does. It reduces the surface tension on the inside of the alveoli and allows the alveoli to expand easily during breathing in. So this idea that I'll just use this alveoli here. You've got this layer of moisture on the inside of the alveolus, which is essentially a layer of mucus, and it's there. Remember what I said it was there for again. It's there. It serves several purposes. At GCSE, you were told maybe that a layer of mucus would trap bacteria, any bacteria and dust that gets into the lungs. Yes, that's correct. But essentially also, the mucus is essentially water. It's watery, and therefore it creates a layer of water to dissolve. So the oxygen that comes in here, there's the oxygen. The oxygen has to dissolve in the mucus, in the layer of water, before it can cross across the oxygen O2, before it can cross across the what? But this mucus, this is a difficult idea. This mucus has what you call a surface tension. Essentially what it means is it's a layer that it has to stretch. So the layer of moisture has to stretch, because when you breathe in, the whole alveoli becomes bigger. The alveoli expands, and so this layer of moisture must stretch. And it's difficult for it to stretch because it's got something called surface tension. But the presence of this surfactant chemical makes it stretch more easily. But if you don't have the surfactant, that means every time you breathe in, imagine your alveoli are like little tiny balloons. Every time you breathe in, the little balloon gets a bit bigger. But it only gets a bit bigger because the layer of moisture on the inside surface can stretch. If you don't have this surfactant, then every time you breathe in, you struggle to breathe in because you have to make your alveoli get bigger. And that's a struggle. And newborn babies have that struggle because they don't have sufficient of this surfactant. What does the question say? State the function of the surfactant and suggest with reference to fixed flow how a lack of it would result in pure gas exchange in premature babies. And you would say something along the lines that the role of the surfactant is to reduce surface tension in the mucus, and that allows the alveoli to expand easily, allows ventilation to happen easily during breathing in. And I just want a wee quick look there just to see exactly what the second mark says there. In question 4, it said it reduces the surface tension in the moisture layer in the alveoli and reducing surface area for gas exchange. So if you don't have the surfactant, then it means the alveoli can't expand, and therefore you have a reduced surface area. So there's the 2 marks. So it reduces surface tension in the layer of liquid that lines the inside of the alveoli, which you'd say is mucus, and that in turn reduces the surface area. So I'm about to finish here. Just having a quick look at what else is in this. What else is in this topic? Okay, so there's quite a few other questions there. Hopefully, you've got the answers. Hopefully, you can have a bash at doing all those questions. And really, you really need to have a go at them all, just to cover all the aspects. I haven't done any practical. Haven't done anything with the respirometer here. But I could do that in a further session or an escape session. So there you are. I think I've covered a good deal of it. I didn't talk about plants and the fact that plants do both respiration and photosynthesis. So I'm looking here in the notes, and I'm looking here actually in the gas exchange section, which is the second half of the notes. I'm looking at this idea that you did at GCSE. Remember at GCSE you looked at the idea that plants would be doing photosynthesis and respiration. In the middle of the night, plants would only be doing respiration because there's no light for photosynthesis. So in the middle of the night, they would be doing respiration, and respiration would release carbon dioxide. So in the middle of the night, plants are producing carbon dioxide. In the middle of the day, they're doing both respiration and photosynthesis. Photosynthesis is using up carbon dioxide. But in the middle of the day, photosynthesis is much faster than respiration. And therefore, the plants are using up carbon dioxide faster than they're using up carbon dioxide faster in photosynthesis than they're making it in respiration. And then this idea that there was some time during the day, dawn and dusk, maybe 6 o'clock in the morning, 6 o'clock in the evening, when the two processes were exactly happening at the same speed. So the plant was now there. Making CO2 and using up CO2, but the two were exactly balanced. And overall, then the plant was observed not to be either releasing CO2 or making CO2. And I haven't gone over that because essentially that was done at GCSE. And you remember, you had experiments at GCSE using an indicator called bicarbonate indicator that would indicate whether the plants were releasing carbon dioxide, making more carbon dioxide than they're using, or so on and so forth. So I'll leave you to look at that yourself. Okay, that's probably as much as I want to do in that. And I can, if you need to, I can do a Skype session at some stage with you. Okay, well done. Hopefully that was of help. All the best. Bye.